Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome everyone to the Genetics Podcast. We have a first ever live episode here from the Flagship Pioneering studios at JPM, and I've got two incredible guests with me today. I've got Michelle Werner, CEO of Alltrna, and Mike Severino, CEO of Tessera Therapeutics. We're going to cover these two amazing companies. They have really big years ahead, and zooming out beyond that, what does it mean for the future of genetic medicine? So Michelle, Mike, thanks so much for joining me.
Thanks for having us.
Pleasure to be here.
I'd love to start with you, Michelle. If maybe you could give people an overview of Alltrna, your approach, and a little bit about the underpinning technology. And I know at Flagship, the part of the raison d'etre is to find a big opportunity, a reason to believe and go for it. So what is yours? And maybe you can introduce this to everybody.
Sure, that's right. So Alltrna is a Flagship Pioneering company that is focused on tRNAs, transfer RNAs. And the way that we see the challenge with rare genetic diseases is the fact that there's somewhere around 10,000 of these different genetic conditions. And with all of the amazing technologies that we have access to, gene therapy, gene editing, mRNA, siRNA, you name it, each one of those technologies really must address one gene at a time or one disease at a time, because that's how those technologies work. When you are a family member that has one of these 10,000 conditions and we're really trying to tackle them one at a time, you can see that there'll be many families that will continue to be overlooked and underserved. And that's actually one of the main reasons why we have so few approved medicines or in the rare disease world. And we're really trying to think differently about what can we be doing to change that paradigm and to really turn it on its head. So this is where the tRNA comes into the picture, and we can talk a little bit more about that in a bit. But with an engineered tRNA, what we are doing is actually, instead of looking at the gene or the proteins, we're actually looking at the mutations that are common across many, many, many different genes. And we believe that actually a single engineered tRNA can be used to address perhaps hundreds of different diseases, which is unlike any of these other genetic modalities. So that's one of the things that we think is so exciting about this technology, because it really may be a great approach to scale innovation across rare diseases in a way that we haven't seen before.
Yeah, amazing. Mike, you also have, I think, a vision to go for a really broad platform-based approach to scale genetic medicine, but using a wholly different approach. I'd love if you could talk a little bit about gene writing and Tessera and your plans for the year.
Certainly. At Tessera, we're developing a platform that we call gene writing. And gene writing is based on the biology of a family of genes called mobile genetic elements. These genes are evolutionarily ancient. They literally go back billions of years in the evolutionary record. And over that time, they evolved various mechanisms to write themselves into genomes. They're so successful at this that they're the most abundant family of genes across the tree of life. You can think of them as nature's genome architects, because half of our DNA as humans comes from sequences that are derived from mobile genetic elements. So when we started the company more than 7 years ago, we asked the question, can we tap into this biology and can we find a better way to write therapeutic messages into the human genome to either cure disease in the case of rare genetic illnesses, or to deliver new instructions, as one might want to do to develop a CAR T therapy, but to do it in vivo instead of at some off-site manufacturing plant with chemotherapy conditioning needed for re-administration. And we've advanced this platform considerably. We have a wide range of families of gene writers that can accomplish different tasks. And we're really excited to take one of those into the clinic this year, as we've made real progress on building this platform.
Yeah, amazing. I think that's actually a perfect segue for where I want to take this for both of you. this is every year is important, but this seems like a particularly pivotal year. You're both going into the clinic and going to test these novel approaches in humans for the first time. I want to just get your perspectives on what you've seen in the preclinical data in the last couple of years as you've been working on it that gives you the confidence in taking it to the clinic this year. And Michelle, let's start with you and then we'll go to Mike.
Sure. So Alltrna also was founded about 7 or 8 years ago. And over the course of this period of time, we've really developed a very robust preclinical data package, especially around our most advanced candidate, AP003, which is the one that will be advancing towards the clinic. What we're seeing actually is precisely to this concept of being able to use a single drug across many different diseases. What we've shown first in an in vitro capacity in the early years is actually using the same engineered tRNA across 25 different disease cell models, different genes that are included, different proteins that are being coded for, and actually mutations that are the same, but also occurring in different locations. And what we have seen in that in vitro capacity is that actually the same engineered tRNA can read through these nonsense mutations, which is what we're targeting in the first instance, and restoring full-length protein. Now, we've also replicated that in an in vivo setting. We've now used the same engineered tRNA in 3 different disease models. We've talked about 2 externally already in a mouse model for methylmalonic acidemia and a mouse model for phenylketonuria, 2 rare genetic liver diseases, which is our first area of focus. And we're seeing really nice protein rescue and, in the same way, using the same engineered tRNA, but different disease models. So it's really speaking to that whole concept. In addition to that, now that we have really advanced substantially through the IND enabling work, we have a very robust safety package that goes along with that. And we are seeing a safety profile that should be very acceptable. It's actually very similar to any other LNP formulated product. So that's also been very encouraging because, of course, with any new modality like a tRNA that has really never been tested before, there's always an uncertainty as to exactly what the safety profile might be associated with the payload. And actually, we're seeing, like I said, a very LNP-focused safety profile versus anything that's specific to tRNAs, which we find really encouraging.
Yeah, it's really exciting. We're going to come back to your unique trial strategy later because I think it's a really important point to think about. The way I think about what you do, you're almost rotating the problem by 90 degrees instead of 7,000 or 10,000 diseases and we need a new molecule for each one. You're taking it and saying this particular stop codon transition, we can treat this potentially across a whole class of diseases. So I think it's an exciting way to think about the problem in a new way.
Exactly.
Mike, you have announced a partnership with Regeneron recently. You also, I think, just got your IND cleared a couple of days ago. You announced that. So again, you've got a super exciting year ahead. What did you see in the preclinical data last year that gave you and now your partners Regeneron the excitement that they have about the program?
Yeah. Well, one of the very attractive features of genetic medicine is that the preclinical models are highly predictive.
Right.
So I'm a physician scientist by training. I've been running R&D in the industry for more than 2 decades. And what we don't like to talk about very much in drug discovery and development is that the models that we use can very often lead us astray. We can build a mouse model that looks like it has multiple sclerosis or looks like it has lupus or some other condition, but it really doesn't. Right. It contains all of the biases that we build into it. And so often insights gained from work in those model systems lead us astray. That's not what we're doing in genetic medicine. We are going after diseases where we know the cause unambiguously. So in alpha-1 antitrypsin deficiency, we know the cause. It's a genetic mutation that, if inherited from mother and father, will lead to this condition. We've known the genetic basis for sickle cell disease, another condition that we're pursuing, for roughly 75 or 50 years or more. We just couldn't do anything about it. Okay. And in the preclinical work that we do at Tessera, what we're doing is we're working in mouse models that contain the human sequence of these genes and showing that we can rewrite them to their intended sequence, to what a geneticist would call wild type. So we know unambiguously that we're getting at the core of the disease. When we can do that in a mouse, and then we carry it forward and do it again in a non-human primate. So the mouse tells us that we can change the human gene in that system. The non-human primate tells us that we can edit in an outbred large animal much more closely genetically related to a human than a mouse is. When we can do both of those things, the probability that we'll succeed in the clinic is very, very high. First principles would tell us that, but we can also look across the field of RNA therapeutics and genetic medicine and say these models have tremendous predictive power, 80%, 90% or greater. That's the equivalent of the predictive power of a phase 3 study in other areas of drug discovery and development. So we get a tremendous head start, if you will, based on the preclinical work. And the preclinical work that we've done on both of the indications that I've described, alpha-1 antitrypsin deficiency and sickle cell disease, give us great confidence that we can write those genetic mutations back to their intended sequence, the sequence that exists in you and me, and offer a real hope for a curative intent for these patients.
And so people understand the underpinning technology in your case, Mike. I think it's interesting, and it's RNA-based, but it is a one-time permanent change, right? And your TPRT program in alpha-1 is I think first in class and the first of its kind. So it'd be interesting if you could talk a little bit about how it differentiates from other programs going. Alpha-1 is a very popular condition to go after because of the clear genetic basis, delivery to the liver, a huge amount of unmet need, a lot of patients, but you have a really novel approach. It'd be interesting to position that relative to other programs.
Yeah. The platform that supports our alpha-1 antitrypsin deficiency program, our sickle cell disease program, and many others like our in vivo CAR T approaches, uses the biologic mechanism that evolved with a family of mobile genetic elements called retrotransposons. And retrotransposons write themselves into genomes. They write themselves into DNA, but they do it through all RNA intermediates. And our RNA-based gene writers do the same thing. So they write new DNA into the genome, but they do it using all RNA compositions. There's a reverse transcription activity that's encoded in these gene writers that allows us to do that. And that's important for a couple reasons. One of the reasons is that the all RNA compositions enables us to use lipid nanoparticle delivery. So gene editing technologies that require DNA delivery today are still dependent on viral vectors, and those viral vectors have many limitations. We believe lipid nanoparticles are a much better delivery solution because they're robust, they're scalable, we can optimize them to deliver to virtually all hepatocytes, and in our hands, based on work that we've done, we can reprogram them to go to other tissues. So for example, in our sickle cell disease program, we reprogram lipid nanoparticles to first avoid the liver, so we detune liver delivery, and then we redirect them to go to the long-term stem cell. And that's what allows us to correct the sickle cell mutation in vivo in the native niche in the marrow where these stem cells live without the need for chemotherapy conditioning or other manipulation. So they're tremendously powerful. And they have a number of other features as gene editors that are quite attractive. We can engineer them to very high efficiency. So we can rewrite a very, very significant proportion of the genes back to wild type, more than 90% in our alpha-1 antitrypsin program in the preclinical systems. And they're very specific and they have very high fidelity. Because there are multiple steps involved in TPRT and each of them imparts specificity, we make the precise change we want to make at the precise location that we want to make it with a very high degree of certainty. For example, we assay rigorously across the genome to look for any unintended edits at locations other than the gene of interest, and we do not detect them, even though we can detect such changes with other technologies that we know have that liability. And so we think that's a real advantage. That combination of LNP delivery, very high efficiency, very high fidelity, making the change we want to make with a very high degree of certainty, and very high specificity, only making it where we want to make it and nowhere else, offer real advantages for this technology.
So I want to talk trials. As we mentioned at the start of the episode, this is going to be a big theme of the year for both of you. Michelle, I want to start with you. One of the things that is interesting to me about your background in particular is you spent a lot of time in oncology, drug development, where basket trials and other novel trial designs are still not maybe the norm, but a lot more accepted and commonplace than they are in rare disease. And you have a really unique opportunity. I know you and your team have been doing a lot of thinking about using novel trial designs like basket trials. I would love if you could tell everybody a little bit how you're thinking about this and the opportunity you have.
Yeah, sure. I think that a tRNA is really uniquely suited to be developed using a basket trial setting. So maybe just to set the context for that, like what is a basket trial? So a basket trial is when you technically have patients who have different disease diagnoses, but the causal mutation of that disease is exactly the same. Patients are identified by having that one mutation, and that they'd be put into one clinical trial, they'd be given the same intervention, and then you'd measure the patient's outcomes accordingly. So as you mentioned, this is a strategy that's used relatively commonly in the oncology space. Actually, I think at least, somewhere around 10 different medicines have gotten approvals using a basket trial strategy. And we're planning to do something similarly with our tRNAs. So for example, as I mentioned, AP003, which is an engineered tRNA, it actually targets a nonsense mutation. That's the arginine to TGA stop mutation, and it's formulated in a lipid nanoparticle. We actually believe that we can do, for example, a basket trial, let's say, of inborn errors of metabolism, all rare genetic liver diseases. We'd identify those patients that have the arginine to TGA stop mutation across maybe 10 or 12 different inborn error conditions. And they would all be given AP003. And what we at least believe would be true, because we've seen this actually in the preclinical data, is that AP003 will work very similarly. These are diseases that have very highly conserved biology. The causal mutation is very well understood. And actually, the biology of the tRNA is also very highly conserved. tRNAs have been around forever. And we're just creating a chemically synthesized version of one, but it's something that the body already recognizes because we have tRNAs in our bodies as it is. So a basket trial might be able to include patients across not only the more common of the rare diseases, but perhaps also these ultra rare inborn errors of metabolism. And one of the real benefits of that, of course, is some of these ultra rare conditions may never have an innovation specific for it, because unfortunately those patient populations are too small where it just wouldn't be feasible to efficiently develop a medicine specifically for that one disease. But by having a development strategy that would allow you to include rare diseases, ultra rare diseases, maybe even nano rare diseases in one trial may actually allow patients to be able to benefit from an innovation that they may not otherwise have access to.
Yeah, that's fascinating. And I feel like it kills two birds with one stone. You open up a much bigger commercial opportunity for the company.
That's exactly right.
But you can also give opportunities to families that would just never have an option. It would never be commercially viable, right? So that must feel very exciting.
Yeah, I mean, no question about it. I mean, coming into this company, it was all about this really could have a profound impact on patients. But it does solve a bit of the challenge that we have with ultra rare disease drug development, is that many companies shy away from those conditions because it's too hard and too expensive to develop a therapy for them. But we're not encumbered with some of those same challenges. So I do think it could be hugely impactful for patients, but then also very beneficial from a financial perspective.
And speaking of unmet need, Mike, in Alpha-1, your lead program right now, you have an opportunity to deliver a one-time, durable treatment that is going to be potentially game-changing, transformative for patients. How do you think about the clinical development plan for here? My understanding from the agreement with Regeneron is you're leading the early phase work, the first trial that's launching this year, and then they're going to take subsequent programs. I would love if you could just talk a little bit about what you're looking for, a little bit about whatever parts of the design you can disclose and how you're thinking about running that program.
Well, one of the very attractive things about drug discovery and development in rare genetic diseases is not only the predictive power of the preclinical models, but the ability to get to answers clinically very, very rapidly. So we're going to run a first-in-human trial that has a typical dose rising design. So you start at a low dose, work your way up to higher doses, with your anticipated therapeutic dose being somewhere in the middle there, based on all the work that we've done preclinically. And we're going to look for all of the typical safety parameters and PK parameters, but we're also going to measure the protein that's produced that's responsible for alpha-1 antitrypsin deficiency. So in this condition, alpha-1 antitrypsin deficiency, when you have two copies of the defective gene, you make an altered form of the protein. That protein actually sticks to itself. It forms aggregates that stay in the liver. And that causes two problems. One, because they stay in the liver, they're not secreted and they can't do their normal job. And what the normal job of that protein is to protect the lungs from destruction as we encounter pathogens in every day of our lives. So that's the first problem. These patients get a destructive pulmonary disease that looks like COPD, right? But advances much more rapidly. It starts younger, it progresses more rapidly, and you don't have to smoke to get it. The second problem that these patients have is because they get these aggregates in their liver, they're toxic to the liver, and they can get a progressive liver disease that can go to end-stage fibrosis and even cirrhosis in these patients. And unfortunately, both of those conditions can be debilitating and life-shortening. So we're going to look at the protein that's produced and circulates in these patients as early as our first-in-human trial. And when we see normal protein, wild-type intended sequence protein circulating, we know we're doing what we set out to do because there's no placebo effect here. You can watch every alpha-1 antitrypsin deficiency patient for their entire lives. They'll never make the normal protein because they don't have the genetic instructions to do that. If you can switch the production of protein from that abnormal form to its normal intended sequence, which we can detect very early on using things like mass spec assays that are highly sensitive, we know we're having our intended effect, and that can really accelerate drug discovery and development. In fact, it should be possible in many major regulatory regions to gain approval simply by demonstrating that you're converting to normal protein at an appropriate level and with appropriate safety and duration of therapy. And so you can move really, really quickly through drug discovery and development in this area. You can go from first-in-human study to approval in times less than 5 years, for example. And that may still seem like a long time, but when you think about drug discovery and development cycles that are 12 or 15 years in most of the industry, it's really quite rapid.
Yeah, are you expecting with the FDA's discussion about plausible mechanism pathways, there might be a path in the US to go quickly?
I think there's regulatory pathways in a number of major jurisdictions, including the US, that can allow us to pursue different approval mechanisms based on the demonstration of features that I just described. Conversion in normal protein, reduction in abnormal protein, ratios of those proteins. And so I think there is, in various jurisdictions, including the US, a lot of flexibility for programs like the ones we're describing.
Exciting. As you think about designing this trial in a competitive field, and this is a question for both of you all because you're bringing novel modalities into cases where there aren't great options for patients, but there are many companies going after these therapy areas with lots of novel approaches. What do you think about from a patient education perspective and from a trial site and provider education perspective, how to think about the medicines that you're developing compared to other either standard of care or potential other trial options that patients might be thinking about? Michelle, maybe we'll start with you.
Yeah, I'd say, the first thing that we're thinking about for our first clinical trial is, number one, where are there higher prevalences of the diseases that we're looking at, and making sure that we have an approach where we can have those countries participate in the clinical trials. That's, I'd say, sort of step number one. The second thing I think is also really important is because these are genetic medicines, and in our case, we're looking for patients that have a certain mutation. We also want to make sure that there's a good clinical practice of genetic testing, really doing that understanding of the full genetic workup for these patients, which really is standard of care, but is not done 100% consistently everywhere. So making sure that that infrastructure is in place. And then the third thing that I would say is really about working extremely closely with patient advocacy groups at a very early age. It's one of the first things that I did when I joined Alltrna 4 years ago, is I started to build relationships with those groups, really understand what their needs are, what they're looking for in clinical trials and new innovations for the patients, what's really going to be meaningful for them, and really bringing them on the journey, really from very early stages to now in the emergence of having a clinical trial. So I think that really helps a lot because they are oftentimes the gatekeepers between companies like ours and the families themselves who may be affected by some of these conditions, and can help with the education around a new modality or genetic testing or clinical trials or all of the above, and then maybe be able to help also with the patient patient identification or to be able to participate. That, as well as obviously investigators, key opinion leaders who also may be treating many of these patients.
Absolutely. Mike, how about you? Same question. How are you thinking about differentiating in a exciting but crowded time for the field?
Well, I think the most important feature of differentiation is the effect that you have, the clinical effect that you have. And when we carry a therapy forward, we always look for cases where there's a significant unmet medical need, where existing therapies are just woefully inadequate. And we benchmark ourselves not only against those existing therapies that are approved and on the market, but against other things that we see in development. And we're always going to go into spaces where we believe we have a real competitive advantage. In each of the areas that we're working, we believe, based on all the preclinical work that we've done, that we can get to really what is the end state. And so what do I mean when I say that? When we're trying to treat alpha-1 antitrypsin deficiency in our preclinical systems, we're correcting 90+% of alleles to the wild-type sequence. Now, this is an autosomal recessive condition. That means you have to have 2 copies of the gene to have disease. 1 copy alone doesn't give you the disease. That tells you that 50 is probably enough,
right?
But we're shooting for 90+ conversion to normal genetic sequence, which would translate to near complete conversion of that protein production that I described to its normal intended sequence. So we want to get right to the end state. We want to make sure there's no need for additional improvement beyond that wherever possible. And we think we have candidates that meet that criterion. That speaks volumes to the treating physicians and to the patient advocacy groups. It really makes a difference. We've shown the data that we've generated to the people who are at the top of their field in terms of treating alpha-1 antitrypsin deficiency, and it just sort of blows them away. And that's what makes people want to participate in a trial. They want to be part of bringing something forward that they believe is going to make a real difference. So beyond that, we're very careful, as Michelle said, with our site selection. We want to deal with sites that have real expertise in this disease and real expertise in bringing forward genetic medicines, because these trials are more complex than a typical small molecule early phase clinical trial. And so when you put all those pieces together, we think that's the recipe for a successful clinical program.
Yeah. What are the key challenges you've anticipated one of my questions perfectly, actually, is genetic medicine trials require a level of expertise that that may not be available at every site. What are the key things that sites need to be equipped with or understand? And the reason I'm asking is because as you all are successful, this becomes what does the healthcare system need to adapt and learn to take on this new technology? So I'll start with you on that, Mike.
Well, first of all, what I would say is the investigators and the staff at the site really need to understand the science behind what you're doing, because they need to be able to explain it to the patients and their families. And if the staff doesn't fully understand it, they're never going to be able to explain it to patients. And these are complex concepts. So you need a site where they're experienced, where they're comfortable with the technology. Obviously, they're not as expert in the technology as the people who developed it, but who can come up to speed quickly and can effectively communicate that to patients and their families so they can make an informed decision about participation in the trial. That's one critical factor. 2, they need to know what to look for and how to manage the typical challenges that might come to play in a therapy of a genetic medicine. They need to have experience managing that. Those are the things that we look for, in addition to just core expertise in the disease area. And of course, centers that treat a lot of the patients of interest.
Yeah. How about you, Michelle?
I would say very similarly to what Mike said. I would say also in addition, especially for the diseases that we're aiming to tackle, inborn errors of metabolism, the standard of care for the most part is medical food, right? A very highly prescribed diet in order to be able to control the symptoms of their disease. So having really excellent nutritional care is really, really critical too, because changes in diet that might be unexpected may also influence the data as well. And so that's something that we're really paying a close attention to when it comes to site selection.
I want to take a moment to imagine that this year's successful and you get that exciting data that you expect to see, and talk about how you scale and expand from here, because you both have so many directions you can take it, and that's probably part of the challenge. But when I think in your case, Michelle, you can go after different codon changes. You can also, I suppose, by tweaking the nanoparticle
go after different organ systems. And Mike, likewise, you think about a different cargo in the gene writer, and likewise lipid nanoparticle changes how you target. Maybe you can just talk at a high level about how you think about these trade-offs between investing in the platform, going after specific indications. And if you have specifics of how you are thinking about it, then that's great. But I know in this business, you have to see the data first and then be agile and make decisions. So Michelle, let's start with you on how you think about that.
Sure, yes. We put a lot of thought into what we call our portfolio strategy. Yes, we are advancing AP003, which is for the arginine to TGA stop mutation in rare liver diseases. We have a second nonsense mutation that we're targeting also for rare genetic liver diseases. So really taking that platform approach, keeping everything else the same besides the mutation. And of course, the engineering of the tRNA to address that specific mutation, but same diseases, but patients that have a different mutation within those diseases. That's the liver program. We could do more from there, but we actually are very excited about exploring the potential of a tRNA outside of the liver. And we actually think a tRNA may be uniquely beneficial to some of them. So CNS diseases and muscle diseases, and I'll give you maybe 2 examples of those. So for example, in CNS, there's a condition that's called Rett syndrome, which is a really debilitating condition. About 30% of all patients with Rett syndrome actually have an arginine to TGA stop mutation. So there's a very high prevalence of these specific mutations that we're targeting, and it's also the one that we know the most about. So
why do so many patients have that particular mutation?
It's a good question, actually. I mean, there must be some kind of a founder. Exactly, right? So that's one that we think really could have a big difference, and there's very few approved therapies for Rett syndrome right now. It also happens to be a Goldilocks disease. And so what that means is that if you actually overexpress a protein or overcorrect, it actually could introduce a different but equally catastrophic condition. Now, overexpression with our engineered tRNAs is absolutely not possible because all we're doing is giving a tool back to the protein translational machinery that it currently doesn't have. So there's no way to get above 100%. So that actually may be a more attractive option for that particular disease. Then there's the muscle diseases. And so, for example, let's talk about Duchenne muscular dystrophy, which of course, as you know, is one that I know very well because I have a child with Duchenne. But it also happens to be a giant gene and a giant protein. And because of that, that is one of the reasons why there are so few limitations when it comes to gene therapies. There are only these mini or micro versions of the gene therapy. An mRNA is just never really going to be able to tackle that size. So a tRNA is actually relatively small in comparison. It's about 76 nucleotides, and you can package it in a lipid nanoparticle or a novel particle. You can do it in an AAV2, or we believe that in the future you can actually create metabolically stable versions of tRNAs and then conjugate and to be able to get to different tissues. So in the case of Duchenne, because of its small size, we actually may be able to use a tRNA, could be the only way to restore full-length dystrophin versus any other genetic therapy. And that really could be a complete game changer for that community. So those are the areas as well from an early stage that we're starting to do some preclinical development to really test some of those hypotheses. And we're really excited about that moving forward.
You also have me thinking about Titin in cardiomyopathy, maybe as another gigantic gene.
Could be, yes.
A challenging example.
That's right.
Mike, how about you? How do you think about, I remember when, so I've had both of you on podcasts previously, and actually Michelle, you twice. I remember from our original episode, you talked about just the vast opportunity that you have. If you think about gene writing, you have full-length genes, SNP changes, the possibilities are endless, right? How do you think about going after that big opportunity?
Well, there really are a tremendous number of opportunities because for the first time, really in the history of medicine, we're able to go after the root cause of disease. And that's just very, very uncommon up to this point. Pretty much the only case where you could ever do that is in infectious diseases.
Right.
But today, we can go after the genetic drivers of disease. We can go after rare genetic diseases. We can go after the genetic drivers of common disease with this sort of technology. And so it's going to be a big year for us in 2026. We entered the year as a preclinical company. Imminently, we'll be dosing patients in our first in human trial, and we expect to end the year as not only a clinical company, but a post proof of concept clinical company that has demonstrated that we can do what we set out to do in patients. And so that's the first opportunity we're tackling. It's a very important one, alpha-1 antitrypsin deficiency. There are between 200 and 250,000 patients living with this condition in the US and Europe alone. And so one can do a lot of good with a therapy like this, but there are many other things we can do. Our sickle cell disease program, which is an in vivo cure for sickle cell disease. So we're not taking stem cells out of the body, harvesting them, sending them off for manufacturing, and then using a stem cell transplant to re-administer them. We're using our reprogrammed lipid nanoparticles to go to those stem cells where they live and do their job in a very protected niche in the marrow. And that's made possible by the technology that we've developed at Tessera. We've also developed the technology to very efficiently rewrite the sickle cell mutation back to its intended sequence. So to allow in model systems today the production of normal hemoglobin where it was not possible, in the very near future to allow that in patients, to allow patients with sickle cell disease to start making normal hemoglobin in a way that would have curative intent. And that's really, really exciting. That program is not very far behind our alpha-1 antitrypsin deficiency program. And there's so many other things we can do. We can use our technology to write whole genes into the genome. We can write CAR T cassettes into T cells in vivo to produce either cancer-fighting T cells or T cells that can help patients with autoimmunity in ways that just aren't possible today. And so we'll be tackling these as we go forward from this year and beyond.
And you could potentially do combinations of things, right? Where you put in a gene and you knock something else out, right? You can really start to think about this as a programmable system, programmable platform.
Particularly for CAR T therapy, for example, we can multiplex our technology. So we can write in a CAR cassette, and at the same time, we can make other changes in the genome. So you could think of, for example, writing in a CAR cassette and then making a change that augments some effector mechanism or knocks out a resistance mechanism that tumors might use to avoid T cell killing. And so we can make very complex and multiplexed compositions with these tools, and that's another one of their strengths.
Yeah, exciting. We're at JP Morgan, and this conference traditionally is all about big deals, fundraising, plans for the years ahead, partnering, and both of you have spent time at the largest of the large pharmas and now leading biotechs. I'd love your perspective on the dynamics right now. There's been a lot of M&A activity in the last year. Great companies like yourselves are successfully fundraising, but it's also challenging for a lot of companies, especially in genetic medicine, where there are questions in gene therapy about what's going to work, what's not going to work. I'd love to just get your sense and finger on the pulse of what it feels like right now, what people could expect for the next year or two. Michelle?
Yeah, I mean, I'd say I think that the tone of the environment seems to be very cautiously optimistic. Yep. I would also say that, in conversations that I've had this week, people have sort of described this as a JP Morgan that sort of feels like a normal JP Morgan. Right. Yes. Maybe not like the last couple of years where it was a little bit more tense, like is the market going to turn? And I think that maybe is a reflection that we have sort of settling into a pace where it seems like things can be picking up, and it could be a healthy environment, which I think is really encouraging. I do think that there are lots of pharma partners that are looking around and are eager to explore new technologies and new modalities and new ideas. And I think that's been great for small companies like ours to see. And I do think that what we're hearing from the investor space, that, people are coming back, that there seems to be a little bit more money in the system to, capital to be deployed. And actually, I think an increased interest in rare diseases. So I'm feeling good about the direction that we're heading this year, and let's see, time will tell.
What do you think is driving the increased interest in rare diseases?
I mean, I think a lot of the things that Mike said, which is, very clear biology of these diseases, very clear understanding of how mechanisms can work. And to be honest with you, I do think that what we're hearing from regulators also is trending more favorably in that direction, and I think that sort of floats boats too.
Yeah, definitely. Mike, how about you? What are your thoughts?
I agree with what Michelle said. I mean, I think that there's a sense of optimism and a forward-looking view at this JP Morgan that may have been absent in recent years based on pressure that the biotechnology industry has been under and just broader macroeconomic and other concerns. And so it feels like things are coming back. But the other thing that I would say is truly differentiated companies make their own luck. And truly differentiated companies can buck trends in a favorable way. And so while we certainly hope that this optimism continues, and we hope that markets can, continue to improve, particularly for biotechnology agents, we're really focused on making our own luck at Tessera, on driving these programs forward, on demonstrating data that have yet to be demonstrated by the field, and really showing that our ambition to have curative therapies for conditions like alpha-1 antitrypsin deficiency and sickle cell disease, can be real and can be real in the near term, and that's what we're focused on.
When you were both having conversations with investors or large pharma partners, what are the key questions that they have about the underlying technology and the things that you're trying to prove out in the next couple of years, Michelle?
So I think the burden to really characterize off-target effects with any new modality is clearly going to be something that's going to be important for investors to understand or pharma partners to understand. In the case of a tRNA, for us, what is the biggest question that we get all the time is, if you can read through the nonsense mutation, the premature termination codon so well, do you also read through the normal termination codon? And is that going to be, off-target is probably the wrong word, but an undesired on-target effect?
Yes.
And so we've done a huge amount of work, actually, where we've kind of put that right in the bullseye of what it is that we're trying to make sure that we characterize and can provide clarity on as much as we can at this stage. And it actually really turns out that biology is in the favor for us, because the context of where a normal termination codon is versus a premature termination codon is completely different. And at the normal termination codon, you actually not just have one stop, but you actually have up to 40 stops all happening right in a row. So biology really ensures that you don't have these elongated proteins at the end of the day. Now, we've characterized this extremely robustly across many, 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 many different genes. And so we feel really confident that this isn't going to be a concern, but the burden of proof is on us to be able to make sure that people feel comfortable with that as well.
Right, because this has been one of the big challenges in the tRNA field historically, right? But it makes sense. So you've got I guess you need to solve the problem at the design level of tRNA, right? And if you can figure out how to recognize that context of this is a true stop codon, or this is one that is aberrant and you should focus on. That's really interesting.
Yeah.
Mike, how about you?
Well, when we think about partnering and interactions with large pharma, I think there are a couple of things that are important. The vision is important, and the vision gets people interested, but ultimately the data is what closes the deal, right? So people care fundamentally about two basic questions. Can you really do it? And can you do it safely? And when you look at opportunities like alpha-1 antitrypsin deficiency, and when you look at sickle cell disease, I think the vision can become quite clear. There's large numbers of patients. These are life-shortening, debilitating conditions that unfortunately involve a tremendous amount of suffering. If you can alleviate that, it's unquestionably a wonderful thing to do, and a very worthwhile ambition. And so then you have to show people that you can really do it. And the data that we've generated that I described in model systems, I think speaks for itself. We can correct with very, very high efficiency. We can do it with very high fidelity, making only the changes we want to make, making them precisely where we want to make them and nowhere else. And we can demonstrate that in a number of different model systems. And that speaks volume to folks. I've run R&D programs around the industry for large pharma for a long time, and I would never bring forward a package sitting in my current seat as a CEO of a biotechnology company that I wouldn't want to be on the recipient of on the other end of that equation. And scientific partnership means a lot. These deals get done when people trust each other, when people trust each other's judgment, when they have confidence in the data that are generated together. And that, I think, is what is really the secret sauce to making a partnership successful.
Yeah. I have one more question for you both, which is one of my favorite questions to ask, just selfishly, because I get to learn a lot from people like you. So I would love to hear your thoughts on a technology or something that you're interested and excited about that isn't directly related to Alltrna or Tessera. So I'll leave it to you to take it as it can be. It doesn't even have to be biology if you really are interested in something else. But I'd love your perspective on something that people may, if we're sitting here in a couple of years, be saying, I remember hearing that from Michelle or Mike, and it's really interesting, and it's gone on to to be really impactful in the field? So no pressure, but what do you have on your mind with that question?
All right. Well, I'm going to say two things, I guess. So you know that I obviously pay very close attention to the Duchenne space.
Yes.
Obviously, that developments in that area could have a direct impact on my family. So two things that I spend a lot of time thinking about is how do you deliver oligonucleotides to muscle?
Yeah.
And so really watching closely what the developments are, from companies that are experts in that area, not ours, that are really trying to solve that problem, tackle that problem of non-viral muscle delivery. So that's number one. Second thing also I think has been really exciting to watch is a lot of the novel exon skipping technology that has happened in the Duchenne space. Unfortunately hasn't been addressing the mutations that are relevant for the mutation that's relevant for my son because he has a very rare mutation in Duchenne, but he could be amenable to that strategy should it, be maybe platformized and can be used broadly across different patients. So I think we've seen huge advancements in that area from a number of different companies that have conjugated oligonucleotides, and I think that's going to be really great for us to continue to, see, explore, and hopefully one day we'll be able to benefit our family.
Exciting. I love that answer. Mike, how about you?
Yeah, it's a challenging question. What I would say is maybe if we zoom out to a little bit of a higher level, what excites me the most is the incredible diversity over what a therapeutic can be in the future. Well, in the present today, and that's only growing in the future. I mean, we used to think about therapeutics as only either small molecules or monoclonal antibodies, and that was it. And there are wonderful therapeutics that are small molecules and therapeutic monoclonal antibodies, but there's still a lot left on the table. If you think about what can be a therapy today, it can be a synthetic transfer RNA, it can be a gene writer, it can be an oligonucleotide, it can be so many different things. And human biology is just so complex. We need a really vast toolkit to be able to interdict the drivers of disease in the way that individual patients need us to do that. And the field is just growing exponentially. And it's really exciting to me to see what we can do today. And it's perhaps even more exciting to think about what we might be able to do 5 or 10 years down the road.
Yeah, I love that answer.
Can I add one more thing?
Please.
Maybe this is also really relevant for your audience. And this is somewhat controversial, but I do think that having universal whole genome sequencing is going to be absolutely critical for the delivery of healthcare of all human beings in the future. Certainly, I would have said that having had access to that in my own family for my son when he was born would have been beneficial for us to have him diagnosed in infancy rather than at 10 years old, and could have changed a lot of the interventions that were available to him. I actually think that all of us would have so much more information about ourselves that would allow us to direct our lifestyles, our treatments, all these different choices in the future. And that's certainly not something we have today, but maybe down the road, I think would be a game changer as well.
Yeah, I couldn't agree more. And I think it opens up the therapeutic possibilities for companies like yourselves if you know years in advance and can design trials in a way that's actually feasible to treat as early as possible, where today we just don't have the early biomarkers in most cases to find patients. So that was going to be mine, actually.
Oh.
I think we're on like the exact same way. Newborn screening in particular is, I think, a macro trend that if we can prove the health economics makes sense in the context of normal healthcare, then it's going to have all these positive externalities. Is there anything you want to leave the audience with? Are you hiring? Are you looking for partners? Is there something you want to leave the audience with? Otherwise, I would just like to thank you both for your time today and for the great work you're doing for patients, which is the most important thing to all of us. Anything?
Yeah, I guess I would just say we're really excited about the year ahead. This will be the landmark year that a engineered tRNA will be first delivered to humans. We're excited about that. So watch this space.
Amazing. Mike?
I think a similar answer. It's going to be a really exciting 2026 for us. I talked about the fact that we entered the year as a preclinical company. We want to exit the year as a post-proof of concept company with multiple programs moving through our pipeline with the opportunity to provide transformative outcomes for patients. And so, watch closely. Hopefully this time next year, we'll have a lot of exciting data to talk about.
Amazing. And thank you, everyone, as always, for listening to the Genetics Podcast. And thank you to the Flagship Pioneering team for hosting this incredible event. It's really exciting to be here.
Thanks for it, Patrick.
Yes.
It's a pleasure. Good.
Thanks, as always, for tuning in to the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy-Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.